In this explainer, we're diving into one of the most powerful biological tools ever created. It's called CRISPR, and it's a technology that literally allows us to edit the code of life itself. So think about it like this. Imagine fixing a serious genetic disease the same way you'd fix a typo in a document. You just find the mistake, delete it, and type in the correct letter. That simple idea, that's really the heart of what makes CRISPR so powerful.
Well, the thing is, that's no longer just science fiction. It's real. Today, scientists can use CRISPR to edit DNA inside living cells with a kind of precision we could only dream of just a short while ago. So let's get into it. What exactly is this technology? How does it work? And what could it mean for all of us? All right, first things first, to really get what CRISPR is all about, we have to go back in time, way back. And the story starts in a pretty surprising place, the ancient, ongoing war between bacteria and the viruses that attack them.
Okay, so the name CRISPR, it's an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. Yeah, I know, it's a total mouthful, but don't worry about the name. Behind all that complexity is a really simple and elegant idea. It's basically a natural immune system that bacteria have been using for millions of years to fight off viruses. Here's how it works. When a virus attacks the bacterium, and the bacterium manages to survive, it does something amazing. It actually cuts out a tiny piece of the virus's DNA and weaves it into its own genetic code. This little snippet acts like a genetic mugshot. It's a memory of the invader. So if that same type of virus ever shows up again, the bacterium instantly recognizes it and can destroy it on the spot. So scientists looked at this natural system and had a brilliant idea. What if we could hijack it? What if we could reprogram it for our own purposes? And it turns out we can. The whole system really boils down to two key parts. First, you have something called a guide RNA. Think of it like a GPS for your DNA. You program it to find one and only one exact location in the entire genome. And then you have the second part, a protein called Cas9. The Cas9 is basically a pair of molecular scissors that goes along for the ride, ready to cut the DNA right at that specific spot. Now you might be thinking, haven't we been able to edit genes for a while? And the answer is yes, but before CRISPR, it was incredibly difficult, super expensive, and honestly, not very precise. CRISPR completely changed the game. It made gene editing easy, cheap, and remarkably accurate. It took it from something only a few specialized labs could do to a standard tool you can find in labs all over the world. Okay, so we've got the components, the GPS, and the scissors. But how do scientists actually put them to work? Let's walk through the process of how this bacterial defense system gets turned into a tool for, well, what's basically molecular surgery. It all follows a pretty clear step-by-step process. First, scientists have to pinpoint the exact genetic mistake they want to fix. Once they have their target, they design that guide RNA to be a perfect match for it. Then, they package the guide RNA together with the Cas9 protein and deliver the whole system into the cells. Once it's inside, the guide RNA starts scanning all the DNA looking for its one and only match. And when it finds it, bingo, the Cas9 scissors get the signal and make a clean cut right through both strands of the DNA. And that cut, that's the key moment. So the DNA has been cut. What happens next? Well, the cell immediately senses that something is wrong. It recognizes the break in its DNA and kicks its own natural repair systems into high gear. And it's right here, in this repair process, that the magic of the edit actually happens. Basically, the cell has two main ways it can patch things up. The first is kind of a quick and dirty fix. It rushes to glue the DNA ends back together. But this can sometimes create a small error, or a typo. This is actually useful if you want to turn off a faulty gene. The second path is much more precise. Scientists can give the cell a healthy piece of DNA to use as a template. The cell's repair machinery then uses this template as a blueprint to perfectly rewrite the broken section, either fixing a mutation or even adding new genetic code. All right, that covers the how. But now for the really big question. Why does all of this matter? How is this amazing tool actually being used out there in the real world, moving beyond the lab and into our lives? And the answer is, the applications are just massive. In medicine, for example, it's already in clinical trials for treating genetic diseases like sickle cell anemia. In the world of research, it lets scientists figure out what genes do at lightning speed. Then there's agriculture, where it's being used to create crops that are tougher against diseases or drought. And it gets even wilder. There are ideas to use it in ecology, like potentially editing mosquitoes so they can't carry diseases like malaria anymore. What all this points to is a massive shift in how we approach disease. You see, for pretty much all of history, medicine has been about treating symptoms. With CRISPR, we're talking about a move from just treating to actually curing, fixing the root cause of a problem at the genetic level. For the first time, we have direct control over the code of life that gets passed down through generations. But of course, a power this big, this fundamental, it's not simple. It brings up some really serious and complicated questions. With all of this incredible promise, there's also potential for peril. And it's forcing us as a society to face some of the toughest ethical debates we've ever had. And here's the core of the problem. Where do we draw the line? I mean, most people would agree that using CRISPR to cure a terrible disease is a good thing. But what about using it to enhance, to make someone smarter or stronger, or to change their appearance? Where does therapy end and enhancement begin? And who gets to decide? And this isn't just some far-off hypothetical debate. It's already happened. In 2018, the world was shocked to learn that the first gene-edited babies had been born. That act was widely, and I mean widely, condemned by scientists everywhere as reckless, unethical, and way too soon. It really drove home just how urgently we need rules and a global conversation about this. So you can really see the two sides of this technology. On one hand, you have this unbelievable promise, the chance to wipe out diseases we once thought were permanent. But on the other hand, you have these huge risks, the danger of creating a genetic divide between the haves and have-nots, the potential for misuse, and the fact that we could make mistakes that are literally irreversible for the human species. When you step back and look at the big picture, CRISPR is a genuine turning point for humanity. For so long, we could only read the book of life. We could study it, try to understand it, but now we can actually edit it. This new ability gives us incredible power to heal and to improve the world, but it also puts an immense responsibility on all of us. Which leaves us with the single most important question. As we move forward, how wisely will we choose to edit?